I'm so excited to skate tonight. Are you? I have this picture of my dog that I think is gonna draw really good.
I got a good picture of Sam, but I also got a good one of her.
Good afternoon, everyone.
Hello. Happy Monday.
It is a Monday. So, well, hope you all are doing well today. Okay, a little business. I will get to grading the exams as quickly as I can. I'm doing my phys exams first because you guys have several things that I've graded already. They have very little that I've graded already, and so I feel it's important for me to get their exams out to them, but I'll get to the exams as quickly as I can. I'm usually pretty good about getting that stuff done. Lab this week. Remember that that's tomorrow. Yeah, the focus tomorrow is going to be working on the outcomes of your projects. So when you come to lab, you should come, make sure you collect all your sensors. Hopefully everybody remembered where they put them. So you can get them downloaded. And hopefully they all worked good. And so the focus tomorrow is going to be on getting your data downloaded, getting it all plotted out, and then sitting down and talking about what the data means. And then if you have time, start writing up your reports tomorrow. But it's a good time. It's a good chance to interact with me on those sorts of things. So you should get your stuff plotted up. We can sit down and chat about them during lab tomorrow. So that's going to be kind of the focus. And then a week from tomorrow, we'll start our second short project where we're actually going to measure metabolic rates, which will be a companion lab for the case study that we're going to start today. I'm undecided. Well, maybe I'm not so undecided. The first lab for our metabolic measurements will involve a lecture that I will give on open flow respirometry because you'll be using it. That will be material that you will need to know for the next exam. So I'm just giving you a heads up warning that you should come prepared to take good notes on that. Anyway, so just be aware that this is not a typical pre-lab, but it's important that you understand how open flow works, and by including it on the exam, I encourage you to really know how open flow works, right? Cool. Anyway, it's a lab you'll have fun with. The first week, we'll measure metabolic rates in crickets, and then the short project will involve measuring metabolic rates in something that you will find to measure metabolic rates in. So that'll be kind of fun. All right, any questions before I get started? He has a Monday face on. Okay, then let's go ahead and start with case study number 2. And I made sure all the sound was plugged in today. All right, this is kind of an interesting paper, Energetics of Burrowing by the ceratulid polychaete. And we all remember what a polychaete is, right? Because we had that covered in our taxon oral reports. Ceroformia morii. And this is a marine polychaete worm that is a burrower. It burrows into marine sediment. And so the authors of this study were interested in reevaluating the cost of transport in these worms. In other words, how much energy does it really require them to burrow into this marine sediment? And generally, cost of locomotion is high for things that have to burrow through solid substances because they have to burrow through solid substances. I would imagine that if every time you walk back to your dorm or house or wherever you lived, if you had to tunnel under the road, you probably would spend a lot of energy, would you say? And just because you're a worm, it's no different. If you have to go through a solid surface, it's costly. So the main goal of this study is to determine whether or not these burrowers in marine sediment, whether they spend a lot more energy on their transport than is spent on other forms of locomotion by other animals. And so you'll see some comparisons when we get to the conclusions between worms and runners, for example. Okay, and the results of the study actually are a little bit interesting and surprising. So the high cost of locomotion in these worms. Now, when you think about it, you know, organisms generally have some amount of energy that they have to spend over the course of the day, and they have to partition this out. And we'll talk about and this is what we call an energy budget. We'll talk about those in a little bit. But the amount of energy that they spend on burrowing through sediments is presumably justified because it's a great way to protect yourself from a predator. Not many things are going to go to the bottom of a body of water and then dig down into the mud to dig you out of there to become lunch. And this is in part because the cost of burrowing to get a burrower would also be high. And there may be a lot less costly things that organisms, predators can forage on. Soft body burrowers, such as these polychaete worms, generally tend to be less efficient at burrowing than hard body burrowers. So if you look at something like a crab, which has a hard exoskeleton, being an arthropod, they generally tend to be able to move through these sediments a whole lot more easily than a worm would, because they don't have to deal with things like body compression. They almost got like built-in tools because they're so hard. If you've ever been to, for example, a beach and seen an Emerita sand crab, if you happen to see one on top, they can just go right through that sediment really, really fast. A worm, not so much, okay? Because they actually have to exert some physical force with smaller parts of their bodies, like their jaws and stuff like that, to get through this solid substance, because again, they're soft body, and that soft body does not give them a whole heck of a lot of leverage. Now, something that's interesting that was pointed out by the paper, and it's actually one of the things that attracted me to this paper for a case study, is that a lot of people, well, let me backtrack and say there's a real challenge to directly measuring the cost of an activity in an organism when that activity occurs within a solid substrate. So when we go to lab in a week from Tuesday to measure metabolic rates, we'll do it in crickets a week from Tuesday just for practice. We're going to put them in a container and we're going to measure their metabolic rate. They're just sitting in air. But if you have an organism that's sitting in mud or some other type of sedimentary material, it's a whole heck of a lot harder, because there are a lot of things that confound the metabolic measurement. It's hard to actually get an instrument into that muck to measure metabolic rate. And so an alternative to this is to measure the forces that are required in external work. And so this is what biomechanicists do all the time. So, I work with biomechanicists on flight, and they can measure the kinematics or movement of things like bird wings and determine how much energy they would have to spend to produce a certain amount of force. So this is all theoretical. And so there are some who have looked at these worms and looked at the external work that they do, okay, measure the kinematics of the various body parts that are required to do the burrowing, and they estimate what the metabolic work has to be, the power output has to be in order to burrow. But the authors are suggesting that a closer look at the mechanical models that the authors used, and we won't talk about those in detail because it just makes all your head spin. It makes my head spin. The metabolic or the mechanical models that are used to estimate the metabolic work that has to be done for this burrowing to occur might not be correct. There may be substantial error in it. And so they are suggesting that the amount of external work that was involved is probably overestimated in some of these earlier studies. And so they proposed actually looking at the cost of transport in these worms by making direct measurements of oxygen consumption, which is something that I do all the time. Now, they will use a different method than what I would use and what we will use in lab. We'll use open flow respirometry. They use an oxygen probe to make their measurement. Oxygen probes are a standard way to measure the changes in oxygen consumption of water or liquids. Okay, and we actually have one lying around here somewhere. I think last time I taught this class, somebody measured metabolic rate, the function of temperature in snails, aquatic snails. So anyway, they're going to directly measure the metabolic work by measuring the oxygen consumption of the worms. And one of the real challenges of measuring oxygen consumption in the mud, besides the fact that it's hard to get an instrument down there to actually measure oxygen consumption, is you get a lot of background metabolism that's going to come from all the microbial activity that is occurring in the sediment. I mean, even single-cell bacteria are going to consume oxygen, okay? And so you have to figure out some way to deal with that. Otherwise, separating that from the actual oxygen consumption of the worms would be very, very difficult. Okay. Another, one of the ways that they are proposing to solve this problem is that rather than having the worms burrow into sediment, they propose having them burrow into gelatin. Okay, and they will make arguments that the gelatin would offer a similar sort of resistance that sediment would, and so they should be able to get a fairly accurate direct measurement of the cause. But the gelatin will not have a lot of microbes growing in it because they wouldn't put any nutrients in there for the microbes. And the other thing that it allows them to do really easily is it allows them to visualize the burrowing kinetics. In other words, the movement, the various body parts or movement, and how they move. So that they would attempt to correlate their direct metabolism measurements with estimates of metabolic power that would come from their kinematic measurements. And so they are going to employ an improved biomechanical model to estimate the metabolic power required to burrow. Does that make sense to everybody? All right. So in this study, the authors are going to directly measure metabolic rate during burrowing and compare it to an improved model that will allow them to estimate the metabolic work based on the mechanical work that the worms are going to do. So that's what we have to look forward to when we get to the results and conclusions of this study. But before we get there, we need to talk a little cellular physiology. We need to talk about energy expenditure. Okay? You all got this memorized? Y'all took Gen Bio, y'all memorized metabolism, metabolic pathways. Well, we're going to review it. Not so sure? Okay, so pathways that are associated with aerobic respiration. Now, if we're measuring oxygen consumption, we can only measure aerobic respiration. And that actually it comes into play. It's pretty important when we get to the conclusions because the authors discover something that's a bit interesting here. So the aerobic pathway consists of three main processes. Glycolysis, which actually is our form of anaerobic respiration. But this connects and becomes aerobic when it connects with the Krebs or citric acid cycle. And both of these feed into the electron transport chain, which is where the vast majority of the energy is actually made. Right? And so we're just going to kind of briefly review these reactions. So in glycolysis, the anaerobic component of all of this, it occurs in the cell cytoplasm. So it occurs outside the mitochondria. And from glycolysis, you get a net yield of 2 ATP and 2 of these carrier molecules that are carrying high energy electrons, NADH+. Okay, these will become important later because these will get sent to the electron transport chain where those high energy electrons will be converted into ATP. Now, if we look at glycolysis, and we're not going to go through here and memorize all the steps to this because you can do that when you take biochemistry or when you took Gen Bio, maybe you learned some of them. We actually are going to spend a couple of ATP up front in prepping, in this case, a glucose molecule for energy extraction. And so we will invest these 2 ATP, and after all of this, we will split our 6-carbon sugar into 2 3-carbon segments. And so the thing that you got to remember is from this point forward, okay, everything in the pathways associated with cellular respiration are going to occur twice. All right. So from here, okay, we have 2 3-carbon molecules that are going to go through the rest of the pathway. Each one is going to produce an NADH+, and each one is going to yield, 2 ATPs. And we will call these in a bit substrate level ATP production. Now remember that we spent 2 ATP up here. So even though we will make 4 ATP, the net yield is 2. All right. So we get a net yield of 2 ATP and 2 NADH from every molecule of glucose. The end product of glycolysis, assuming that we are functioning aerobically, is going to be pyruvate. And so 1 molecule of glucose will produce how many pyruvates? 2, yes. See, you're all listening today. Okay, and then pyruvate, if I can get my mask back around my ear here. Then pyruvate will combine with an acetyl group here to produce acetyl-CoA. And then the acetyl-CoA will enter into the Krebs cycle. Now, we get 2 NADHs that are going to be produced from each of the pyruvate, 1 from each of the pyruvate molecules that will enter the Krebs cycle here. And the other thing that gets produced here is carbon dioxide. And the reason why I'm pointing this out is because during energy production, you're not only going to consume oxygen, which only happens in one place in all of this, but periodically, you're going to produce some carbon dioxide as well. And this is one of the steps where carbon dioxide is produced. The amount of carbon dioxide that is produced during the breakdown of metabolic substrates that you are breaking down for energy, such as glucose, but it could be fat or it could be protein. The amount of carbon dioxide that is produced is going to be dependent on the type of metabolic substrate that you are consuming for energy. So you'll get a different amount for sugar than you will for fat or for protein. And that will become important in a little bit. So the Krebs cycle, as I mentioned, turns twice for each glucose molecule. And as we go all the way around here, we have several energy producing steps. Okay, we produce 2 FADH2, which is another carrier molecule that will carry high energy electrons. Okay, and that step is right here. And then we will produce 6 NADH plus. We have a step here, we have a step here, and we have a step over here for that. And then we have one, what we will call in a moment, substrate level production of ATP, of energy in this pathway as well. And we also produce CO2. So we have some CO2 that is coming off here, and we have some CO2 that is coming off here, right? So those are areas where we have CO2 production that is associated with the breakdown of our metabolic substrate. Questions at this point? All right, so here is just a summary slide of all of this. So this just summarizes everything that we get from a molecule of glucose right here. So we have our net 2 substrate level ATPs produced. We have 2 NADHs, which are going to yield 4. That's kind of an odd number for NADH, because we usually get 3 for each NADH, but this particular one only gets 4. So here's 2 more NADH that we get 6 ATP from, another substrate level ATP production, so we get 2 ATP from that, 6 NADH from the Krebs cycle, so that's 18 ATP, 2 FADH, because it actually enters the electron transport at a different place than NADH does. We only get 4 ATP from that. Add them all up, and you get 36. Now that number could vary a little bit, because when and this is where this is kind of weird, because remember that glycolysis occurs outside the mitochondria in the cytoplasm. So that NADH has to be transported into the mitochondria. And that transport involves transferring those 2 high energy electrons from one transporter to another. And that other transporter can either be an NADH or an FADH. And so that's really what dictates what's going on here. They're assuming it's going to be picked up by an FADH here. But in reality, it could be another NADH, and this number would turn to 38 if that was the case. So a lot of times on these diagrams, you'll see it listed as 36 to 38 HP, depending on what happens to these electrons right here. But that's not important. The key thing here is when we're functioning aerobically, we get a whole lot more bang for our glucose buck than we do if we're only functioning, anaerobically, which your net gain in ATP for that same molecule of glucose is only 2. Y'all with me? All right, let's talk for a moment about the electron transport chain. The electron transport chain is really interesting because it functions, most people don't really understand how the ATP get produced in the electron transport chain, but it's really interesting. So here we have where NADH is dropped off right here. And then FADH is dropped off where is it? It's not even listed on here. Am I missing it? I don't see it. Anyway, it's dropped off a step later than the NADH is. which is why it doesn't produce as many ATP. And what these high energy electrons do as they move down the electron transport chain is they're not directly producing ATP, but what they're doing is they are powering the movement of these protons from one side of the intermitochondrial membrane to the other. So to the intermembrane space, they're pumping all these hydrogens. This is basically a form of active transport. They are creating a really huge proton gradient in that intermembrane space in the mitochondria. And then what that hydrogen does is it's going to go through this ATP synthase right there. I think I got a bullet point for that. And it's actually this ATP synthase that produces all the ATP. Now, if you ever are studying thermoregulation, and Ian will remember this because we mentioned this in physiology, you have these things that are called uncoupling proteins that a lot of organisms will use to produce heat. Some ectotherms will actually produce heat internally using these uncoupling proteins. In physiology, we talked about how hummingbirds produce a bunch of these uncoupling proteins in order to warm themselves up when they're coming out of hypothermia, deep torpor. And what these uncoupling proteins do is they basically stop this so that the energy from this proton gradient, instead of going to make ATP, it just used to produce heat. They basically take advantage of a high level of inefficiency in this biochemical system to generate a lot of heat to warm themselves up. And so there are some proteins that actually will take advantage of just altering this process. All right, questions? Y'all still with me? All right. So I'm just showing you this diagram right here to just to make the point that we've talked about energy production with regard to glucose, which is the way everybody pretty much does it. But also fats and proteins will feed into these pathways. And they feed into them a little bit differently, which is why we get different differential amounts of carbon dioxide produced. But the fats and proteins can make their way into the Krebs cycle and be broken down for energy. All right. The other thing that I will mention from this is you'll see here lactic acid. If you are functioning anaerobically, then what you will produce from that pyruvate is going to be lactic acid. Now, the beauty of this system is that this is a reversible reaction. So you build up this lactic acid, you can back convert that to pyruvate, and then the pyruvate can make its way through the Krebs cycle. And so when you create an oxygen debt, that's basically what you were doing. You're building up a high level of lactate in your system, which can do things like make your muscles sore. But then when you are at rest and you are repaying an oxygen debt, you can basically convert all that lactic acid back to pyruvate, and then it will go and you'll reclaim the energy that you temporarily lost. One of the, you know what, there was one other thing I want to say about this that I just totally spaced on. This step right here in the electron transport chain. 2 hydrogens, basically 2 electrons, plus a half O2 creates H2O, metabolic water. That step right there is the sole reason that you breathe oxygen. It's the only place where oxygen plays any role in this entire process. But without the oxygen, basically to pick up those electrons at the end of the electron transport chain, it all gets shut down. The whole thing except for glycolysis gets shut down. And even glycolysis can only serve as an anaerobic pathway to produce energy at that point. And no endotherm can survive for very long on just the anaerobic pathway. The other thing that I will say about that step is a lot of organisms that live in arid environments actually make good use of the metabolic water that is produced at the end of the electron transport chain. So if you look at something like a kangaroo rat that lives in a desert environment, Knut Schmidt-Nielsen many years ago did some measurements on the amount of metabolic water that they produce and discovered they can meet a huge portion of their daily water budget through the production of metabolic water. And a lot of organisms will adjust their diets in order to maximize the net gain in water. The net gain in water that comes from metabolic water, particularly in places where they are going to be water stressed. So for example, the last thing that you want to consume in large amounts in your diet, and I'm getting a little tangential here, but this is kind of interesting since we're talking about it. The last thing that you would want to eat a lot of if you are depending on metabolic water is protein. Because protein requires a lot of water to excrete. You create a lot of toxic waste products when you are breaking down proteins, and you excrete a lot of that in your urine. And so that requires a lot of water to get rid of. And so that drops the amount of net water gain you get from a process like metabolic water production. And so what you'll see is a lot of organisms will shift their diets. And this would be true of an insect that lives in an arid environment as well. They would shift their diets to something that is going to be, probably something even fats are going to be better. You get a lot of bang for your buck with metabolic water when you're consuming fats. But fats or sugars are going to be better than protein because they require less water to excrete. Okay. I want to talk for a moment about the respiratory exchange ratio. This is why I was making a big deal out of carbon dioxide being produced. You can learn a lot about the metabolic state, well, maybe metabolic state. Yeah, I suppose that's a fair word. The metabolic state, or learn a lot about what an organism is burning for energy by looking at the respiratory exchange ratio. Now, this table right here says RER and RQ. When I was your age, RER was not a thing. Nobody really even used the term respiratory exchange ratio. Everybody talked about respiratory quotient. But there's a difference between respiratory quotient and respiratory exchange ratio. But first, let's talk about how you calculate both of them. You calculate both of them simply as the ratio of CO2 produced to oxygen consumed. The difference between RER and RQ is RER is that ratio at the level of the lung. So if I exhale air, I can measure what my oxygen consumption was, knowing what the oxygen content of air is, and I can know what my CO2 production was by measuring the CO2 in the air and comparing that to ambient air. Okay. So So I measure that at the level of the lung. So what's coming out of my mouth when I exhale, or what's coming out of a spherical, if we're talking about an insect. Because I got to remember this is inverted, right? RQ is the same measurement, but at the level of the cell. Now, if you look under RQ here, you're going to see three numbers. You're going to see that this ratio is 0.7 for fat, 0.8 for protein. Now, that the protein value strongly depends on what kind of an organism we're talking about. Okay, I'll explain that in a minute. And then 1 for carbohydrate, which means if you are burning carbohydrate, you're producing, for every oxygen molecule you consume, you're going to produce 1 CO2. All right? The range will always be from 0.7 to 1 at the level of the cell for the RQ. That is not the case for RER, because this range of 0.7 to 1 assumes that an organism is at steady state, at equilibrium. If an organism is not at equilibrium, and so the example they give here in this table, during exercise, you're not at equilibrium during exercise, then your RER can actually exceed 1. And that's because you are producing lactic acid. You've got all kinds of bicarbonate stuff going on in order to buffer that increase in acid in your system. You're making more CO2. So you can produce a lot of CO2. Okay? And so your RER can be above 1. Right? So the whole existence of RER was created just to deal with this, with the fact that what's coming out of your lungs when you exhale, it often does not represent an equilibrium state. Now, protein. This 0.8 value for protein is only valid for organisms that produce urea. If you are an organism that produces uric acid, for vertebrates, that would be birds and reptiles. But there's a lot of insects, okay, a lot of arid invertebrates that produce uric acid because it is the best, it's the best byproduct of protein metabolism to produce if you're trying to conserve water. So if you are a uric acid producer, then the value, the RQ value and the RER value, if you're at steady state, is closer to that for fat. So the value for fat is something like 0.71 actually. The value for protein for a uric acid producer is 0.73. So it's really close. So when you're looking at what this ratio is for an organism, and you want to know the protein value, you got to look at what what it excretes after, when it metabolizes protein. Questions? I just threw this on here because it just talks a little more about things that can drive up, change CO2 production. So RER at the lungs can be greater or less than RQ at cell. It can range from slightly below 0.7 all the way up to around 0.5. So they offer as an example here hyperventilation, which would represent exercise, and hypoventilation, which would represent a rather low metabolic rate, possibly even hypothermia. But hyperventilation is known for just blowing off a lot of CO2 really fast, and so that can get your CO2 content way up. Other things that will throw off RQ if you are, or RER, I should say, if you are starving. So you go off and you start metabolizing yourself, basically, for energy. If you are depositing fat, that's not an equilibrium state when you're storing fat. So that also will throw your RER off. So anything that takes you out of that steady state mode. Here are just some real numbers. So protein, oxygen consumed, almost a liter. CO2 produced, about 0.8 liters. So you get an RQ value of 0.8. You can just walk down the rest of these here and see how the ratios are produced. And then, anyway, any questions about RQ versus RER? This is a good tool if when you do your short projects and you have some interest in knowing, say, what some invertebrate that you're interested in, what maybe they're metabolizing for energy. You can use a measurement like RER in order to assess that. So, I work on hummingbirds, it's not an invertebrate, but, I've done some measurements on torpor where you measure their RER at the start of the night. It's very close to 1. And then after a couple of hours, you can see it drop down to something that is more appropriate for fat. So that tells you a little something about what metabolic substrates they're using to power their nighttime metabolism. So there's a lot of uses for this type of a measure. Okay, that was a little tangential relative to the case study, but so let's get back to some things that are going to be directly related. Let's talk about energy budgets. So one of the reasons why the authors of our case study are interested in this cost of burrowing is because most organisms have a certain amount of energy they can spend over the course of a day. And they have to proportion this out in terms of its use in ways that are going to promote their survival. If you spend too much energy on locomotion, don't have enough for maintenance, then your body's going to deteriorate. You're not going to survive very well. And so all organisms have to manage their energy budget. Okay, how do they divide up the pie in order to make sure all the necessary things get powered by energy? So here is a penguin. Yeah, I know it's not an invertebrate, but it's the best. It's the best diagram that I could find. Not many people make diagrams that involve invertebrates of this sort. Go figure. So anyway, all organisms are going to ingest energy in some chemical form. I should say all animals. Okay. Not plants, although some do. Okay, so chemical energy is going to be ingested, and then that energy is going to be shunted into a variety of categories of use. And so I've listed 3 of them here that we will go through. So the first one we'll talk about is biosynthesis. So the chemical energy is absorbed, and then that energy will be used to promote the building of more complex molecules. So this could be storage sugars, so polysaccharides. This could be fat. This could be proteins. This could be nucleic acids. It could be a host of different things. Anything that your body is synthesizing. Okay. Now, the key thing to remember about all biochemical processes, they are all subject to the laws of thermodynamics, and therefore they are not efficient. When you look at the overall state of living things, we all tend towards entropy. Okay? And so when we look at biosynthetic reactions or any type of a biological reaction, it's not going to be 100% efficient. Some of that energy is going to get converted into other types of organic molecules. And I named several of them. But a chunk of that energy is just going to be lost as heat because the synthesis is not 100% efficient. And so biosynthesis is going to be important for things like growth, energy storage, and absolutely reproduction. So if I am managing an energy budget and I'm having, a tough year, maybe I don't grow, maybe I don't store a lot of energy, and maybe I don't reproduce. And so I can cut back that whole category at some level is an optional, it's an optional form of energy use for animals. Because if they don't have the energy to spend on it, they can shunt a little more energy to processes which are necessary for survival. Right? Or maybe they store energy but don't grow and don't reproduce, just so they can make it to the next year. Now, if you are a short-term reproducer, that may not be the strategy you use. If you only live for one cycle, then you don't store energy. You dump everything you can into reproduce and try to get reproduction done before you die. And so the priorities are going to depend on what the life history for the animal looks like. So anyway, that's biosynthesis. Then there's just basic maintenance. So maintenance is going to involve things like basal or standard metabolism. For everything that we study in here, it would be standard metabolism. These are measures of the minimum amount of energy that is required to run just the basics of your physiology. Just the minimum to keep you going. So the closest that any of you guys ever get to basal metabolism, because you're into things, would be when you sleep. That's your lowest level of energy expenditure. You're just keeping everything on that has to be on. SMR is just like BMR, but for an ectothermic animal. And the difference between BMR and SMR is temperature dependent. Because ectotherms do not, and all invertebrates are ectotherms, they do not internally regulate the body temperature. They have to do with behavior. And so if you want to know what that minimum metabolic rate is, you also have to specify a temperature. Now, what kind of things do you have to think about when you are measuring SMR in an organism? You have to make sure that the organism doesn't have anything going on that would add to those costs. So to make those measurements, we generally measure it on a fasted animal because digestion costs energy. We generally measure it in the dark because being in the light promotes activity. You want it to be at rest. We generally make the measurement during a period when the animal would be resting, i.e. sleeping. I don't know what that would look like for an invertebrate, but whenever, perhaps at night, when it would just be quiet. If we were talking about an endotherm, we would make it within a range of temperatures that we call a thermal neutral zone, which is a range of temperatures in which you spend no extra energy on temperature regulation. But that's not applicable to an invertebrate because they're all ectotherms. And so we would specify what temperature we were making the measurement at. It's actually a very difficult measurement to make. A lot of times we will opt to measure what's called resting metabolic rate instead, because we don't have to confine ourselves to these restrictive conditions. And we just try as best as we can to measure the animal at rest. And then we define what we mean by that. Then we report the results. So that's basically maintenance. Again, there's going to be some inefficiency that's associated even with maintenance, and there will be some heat that is produced, lost, because of the inefficiency of the reaction. I need to finish up real quick here. External work is the big one, because muscles are not very efficient. And you work hard, you go out and exercise, you sweat. And that's because the activity of your muscles generates a lot of heat, because muscles have really poor mechanical efficiency. You know, often in the neighborhood of maybe 20%, maybe even less. So you produce a lot of heat for energy that you are shunting to do mechanical work that involves muscles. Okay. And so that will also produce a fair amount of heat. Okay. All right, so I'll stop there. We'll pick this up on Wednesday.
Are we gonna finish the lecture with time to do or lightning on Wednesday?
I'm thinking lightning talks will probably happen on Friday.
Okay.